you seen, na-mention ko na ba class last time na there is now a notification about the evaluation form that you have to accomplish. Na-mention ko na ba yun sa inyo? Please kindly accomplish the evaluation form, evaluation of teaching performance, no? For learning term 1. Ang biochem is offered sa tong learning term 1. So you have to accomplish the form um, both for LEC and lab. Okay? And then, may due date dito na September 24. But sa akin, dapat earlier, September 22. So today, September 21. Tomorrow, uh, 22. Basta before 24, no? Kailangan no? So please, when you accomplish the form, okay, uh, tapos, of course, you have to submit it. Meron dapat, a proof that you have accomplished and submitted the form. Have a screenshot of that and then submit to me. I will make a page, no? Sa announcement, I'll make a page wherein you have to submit the proof. Okay? Nagkakaintindihan ba tayo doon, class? Kailangan kasing medyo pukpukin kayo kasi kami rin pinupukpok kapagka hindi nag-e-evaluate yung mga students namin. So, nakaano sa amin yun, naka-reflect kung ilang students lang ang nag-accomplish ng evaluation form. So, if you submit the proof dun sa page na gagawin ko, then you'll get an additional 10 points for your CP. Okay? Am I clear on that, class? Am I clear on that? Hello?
Yes po.
Have I also mentioned that, kasi hindi ko, for your CP points, there are additional points, right? Okay, so what are my additional points? One is for your attendance, okay? Siguro 10 virtual classes na tayo, no? Kasi this is our 12, yung last time 11, ano na yon, kasama na ng final grading period. So I think mga 10 virtual classes. So kapag you have 9 to 10, ibig sabihin, siguro kung wala, isang beses wala ka. Okay, so basta you have an attendance of 9 to 10, okay? So you'll get an additional points of 10 for your attendance. Okay? So your attendance plus that evaluation form is already 20 points. And then, di ba may mga recitations tayo? Okay? Halos lahat naman natawag ko pero others talagang hindi sumasagot. So, if you have recitations of 2 or more, then you'll get another 10 points. Kapag once lang yung recitation mo, then you'll get 5 points. Kung walang recitation, so you have no additional points for recitation. So therefore, a total of 30 points will be added to your CP. Ibig sabihin, class, no? So what you will do is, nakikita nyo naman yung mga scores nyo. You see your scores for each activity, no? Meron nga lang na 3 activities na hindi ko pa na-checkan. Okay? So, 3 na lang. So, basta by this week, matatapos ko yan. Okay? So, what you do is you simply add your scores for all the activities. Of course, do not include the midterm score. Iba kasi yung sa midterm score. Basta yung sa CP lang, all the activities. Okay? Yung kasama yung mga postas, ganon. So, you simply add your scores there plus yung additional points. So, alam nyo naman kung nakapag-recite kayo at saka you know if you have a complete attendance and of course, kung nakapag-evaluate ka, you submit the proof. So, you simply add whatever the points, additional points you have to your score. Then, over the total score. Yung total score, yun lang points dun sa mga ah, ah, activities. Okay? And then, yung um, times 100, so you get a percent score, then I will send a transmutation table where you can see the grade equivalent. Okay? And then, after that, pag nakuha mo na yung grade equivalent, so how do you combine the CP and the periodical exam? Yung periodical exam nyo, ganun din. Since it is 100 items, 100 points, no? Whatever is the score you got, raw score, yun na rin yung percentage score mo. And then, titignan mo na lang din uli dun sa transmutation table, yung grade equivalent. And then, your CP, for example, if that is, you got 2, times 2, and then yung periodical exam, kung ang nakuha mo is, sabi mo ng 1.5, so times 1, and then you add the 2 divided by 3, that's your left grade. Okay? Anyway, sa susunod na kapag natapos ko na lahat checkan, then we can compare no whatever you have computed with the grade template, no? The computed ones using the grade template of CEU. Okay? So, hindi naman ako ang magko-compute, kundi ipapasok ko lang naman dun sa grade template na ginagamit namin. Okay? So, now, ano pa? So, don't forget that, ha? Kailangan, basta ano, uh, dapat 21 ngayon, dapat 22 bukas, no? Or hanggang Saturday, no, mai-submit nyo yung proof. Okay? No, ano pa? Ano pa bang uh, anything that uh, kayo, meron ba kayo gustong itanong before we proceed to our discussion for today? Class, meron ba? Uy, yung mga new activities, wala pa akong nagagawa for the new activities. Okay? So, hindi ko pa maano yun. I'm still, ang ginagawa ko is checking, no? Yung manual checking as well as, of course, preparing for the discussions. Okay? So, para sa mga activities for enzymes and nucleic acid, hindi ko pa nagagawa. So, siguro next week na natin i-due date. Okay? So, any other question? Hello? May I download the attendance? Sandali lang. Oh, yeah. That's the first attendance, and we are 41 users. Okay. So, okay. Of course, 7 o'clock ang klase natin. Siguro marami pa sa atin na hindi nagbe-breakfast, right? Okay? 'Di ba? But, of course, siguro naman, kahit papaano, nagwisik-wisik ng konti, 'di ba? And siguro you have seen yourself, no? Siguro naman may nagsalamin na, 'di ba? Right, class? Have you seen yourself, your reflection or your image on the mirror, class? Ha? Ah, ano bang napansin today? Ha? Every time that you look at the mirror and see yourself, what comes on your mind? Anong naiisip? May I hear an answer from anybody, class? Ay, sandali nga lang. May I get my, ano? Okay, my record, no? Record book. Okay. Oh, Miss, oh yes, yes, Miss Cruz. Miss Cruz, sige nga, ikaw na. Miss Cruz. Yes,
Good
Miss.
morning po, ma'am.
Yes, good morning. Nakita mo na yung sarili mo ngayon. Nagsalamin ka na ba?
Opo.
Opo.
Kanina po nung nag-picture po,
Ay, yes, of course. Okay, 'pag nakikita mo yung reflection mo, ano yung pumapasok sa isipan mo? Sige nga,
um,
share it with
medyo
us.
puffy pa po yung mata kasi halos kakagising lang din po.
Okay. How about, do you have any, uh, yung parang wish, okay? You look at the features of your face, okay?
Siguro po ano,
No.
maging clear skin po kasi medyo nagbe-breakout po ako ngayon dahil po exam week po last week. So, napuyat po.
Napuyat, okay. So, worry tayo, minsan nagwo-worry tayo sa how we look, right? And sometimes, okay, minsan iniisip din natin yung 'pag merong hindi medyo okay sa features natin, "Uy, pumasok ba sa isipan niyo na, sana medyo maganda ito, sana nakuha ko yung ilong ni papa," yung gano'n. "Sana nakuha ko yung mata ni mama, mas maganda kasi." Does that come to your mind?
Minsan
Miss
po, ma'am.
minsan po. Pero of course, wish lang naman yun, 'di ba? We still love what God has given us, right? We still cherish what we have and we just pwede naman i-improve, right? Pwede naman i-improve. But what
Yes
do you
po.
think? Yes. Okay, next. So that's Miss Cruz for the first recitation ng ano na natin 'to ha, ng ating final grading, period. Okay, Miss Ancheta, okay, may I hear from you, what do you think, okay, what do you think, okay, what are responsible you? What are responsible for those features na nakikita natin sa ating face, okay, what biological molecules kaya that are responsible for the features that we see sa ating katawan, sa ating, especially sa face natin, kasi yun ang madalas natin makita eh. Yes, Miss Ancheta. Ancheta ba?
I think po
Yes.
ano, yung DNA po natin napasok
Yes,
po
the
sa preference.
DNA. Yes, tama. The DNA. And we know that uh, there are informations in the DNA that are passed on from our parents to the offspring, to us, right? Okay, now, okay, what are in the DNA? Okay, okay na si Miss Ancheta. Okay. Next, volunteer. What are in the DNA? What specific biomolecules are in our DNA? Yes, Miss Kapili, the highest. Miss Kapili, yes.
Ma'am, is it nucleotides po?
Nucleotides, yes, or nucleic acid. So this is our topic for this for today. Chapter 22 of Smith talks about or discusses the last biomolecules or the last macromolecules, no, which are the nucleic acids. Okay, so those ano, no, features, okay, that we see, okay, we have these features, unique characteristics, okay, and these characteristics are determined by the nucleic acid polymers that reside in the chromosomes of our cells. Now, where do we see the chromosomes? Nasaan ba ang chromosomes? Another hand, please. Where are the chromosomes? Nasaan makikita? In which organelle of the cell? Our cell. Yes, Mr. Ku. Mr. Ku, ikaw na, Mr. Ku. Ku?
Nucleus po.
The nucleus. Yes. Okay. Ayan. Yan. Yes, of course, no, in the nucleus. So, nucleic acids are unbranched polymers, just like carbohydrates, proteins. They are naturally occurring polymers. Okay, and of course, kaibahan, no, unbranched. Okay, they are unbranched. Even proteins, unbranched ang proteins, di ba? But carbohydrates, may branches yan, okay? So we have unbranched polymers. And the repeating monomer, okay, is called the nucleotides. Okay, so we have nucleotides as the building blocks of nucleic acids. And there are two types of nucleic acids, the DNA and RNA. Okay? Now, the DNA, also called no, abbreviation for deoxyribonucleic acid. This one, okay, stores the genetic information of an organism and transmit that information from one generation to another. Okay? So kaya 'yan yung ano no, yun yung responsable doon sa nakikita nating mga features ng ating katawan, okay? And then we have the RNA, ribonucleic acid. This one translate the genetic information contained in the DNA into proteins needed for all cellular function sa lahat ng pangangailangan ng ating katawan ng proteins. Okay? So, yun yun. And so, mhm. um, and just like sa proteins nung pinag-usapan natin, yung sequence ng amino acid doon may effect sa atin kapag naiba, 'di ba? Ganon din sa DNA. If there is even a slight alteration in the nucleic acid sequence, okay, that can have a significant effect on organism. Since yung nucleotides na nandyan sa DNA which carries specific bases, okay, three bases that codes for a specific amino acid. So, pwedeng anumang alteration doon can lead to serious diseases. Like for example, yung sickle cell anemia na pinag-usapan natin sa proteins. Okay? So, now before let's discuss muna the structural aspect of this um, nucleotides, okay, the building blocks of nucleic acid. So, the nucleotide monomer that compose the DNA and RNA are composed of, okay, tatlo yan. You have the monosaccharide. This one is the monosaccharide. Okay, it's a cyclic structure. From the structure, we can see that it has one, two, three, four, five carbon. So it's a pentose. Specifically, it's an aldopentose. Okay? And we have also learned from topic carbohydrates that, okay, the carbon, okay, right after the oxygen, this one, we call this the anomeric carbon. Okay? The anomeric carbon. In the open chain, it is the um, a, a carbon bearing the aldehyde group, okay, or the carbonyl group. So ito yon. So this is the anomeric carbon that is bonded to the second component. What is the second component? It's a nitrogenous base, nitrogen containing base. Pag tinignan natin ito, okay, may mga nitrogen. So this is basic. So it's a base. So nitrogenous base ang tawag dito. At there are different types of these nitrogenous bases. And then on the left side of the aldopentose or the ano no the monosaccharide, which is ribose, by the way, no, five carbons, ribose, you have another component which is a phosphate group, and the phosphate group is attached to where, to the fifth carbon of the monosaccharide. So one like this is a nucleotide. Okay, now next. Mhm. So kanina doon sa first slide, na-present na kagad yung the first the difference between DNA and RNA. Okay? This one, another differentiation between DNA and RNA. DNA molecule contains several million nucleotides. So yung kaninang pinakitang one monomer of nucleotide, so a million of that, several millions of that, no? While an RNA molecules have only a few thousand. And then, where do we see the DNA? It is contained in the chromosomes of the nucleus. And each chromosome having a different type of DNA. And bawat species may kanya-kanyang number of chromosomes. Tayo, human beings, have 46 chromosomes, 23 pairs. And each made up of many genes. Pero ano ba yung gene? A gene is the portion of the DNA molecule responsible for the synthesis of a single protein. But class, hindi a portion lang yun, maliliit na portion lang yun. It's about 1 to 2% lang of the whole DNA molecule codes for a single protein. The rest, they are non-coding. Ibig sabihin, not converted into proteins. Okay? And so, what else? So this is, uh, dali lang. Okay. Mhm. So this is the very, very large, no, enormously large DNA molecule, okay, that compose the human genome. Ano yung human genome? That is the total DNA content of an individual. Okay? And these DNA molecules is packed, ito yun, packed tightly. Okay? Packed tightly, ito yun. Packed tightly into the nucleus of the cell. So this is the nucleus. This is the cell, this is the nucleus. Okay? And the double-stranded DNA helices. So yung DNA molecule natin, class, is made up of two strands, two polynucleotide, okay, that are wind around each other and then that twisted with each other, okay, and then wind around a core of protein molecules called histones. Ito, these are protein molecules. This is called histones. So naka-wrap around dyan yung double helix. Yun, double helix DNA. And so a chain of this, you form nucleosomes. Okay, ito, nucleosome, a chain of nucleosomes. And the chain of nucleosomes wind into a supercoiled, nakawan yan, supercoiled fiber called chromatin, okay, which composes each of the 23 pairs of chromosomes in the human cell. Okay, so that's how we see the DNA molecule. Okay, so now, tignan natin naman yung pagkaka-join ng mga components of this nucleotide. Okay? So, we have here what we call nucleoside. Ano ba yung nucleoside? Nucleoside is a monosaccharide joined to a base. So, yung dalawang components muna. Okay? Dalawang components ng nucleotide. Yun munang monosaccharide and a base. So, another differentiation between RNA and DNA, the RNA, the monosaccharide is the aldopentose deribose. This is it. Okay, and um, uh, the capital R in the RNA that represent D-ribose, naka-red yung color, 'di ba? So this is D-ribose. And for the DNA, the sugar component is the 2-deoxyribose. So letter small d yun. Yun yung capital D ng DNA. So sa quiz, if ever na tinanong kayo na ang sagot is DNA or RNA, you have to write this in capital letters. Okay? 'Di ba? Pag sabi natin pag acronyms, capital letters yun, ha? Okay. Oh, next, what's the difference between these two sugars? Of course, the D-ribose, pag tinignan natin yung this is the carbon 1, carbon 2, okay, you have an OH group, okay? A hydroxyl group attached on carbon 2. Doon kay 2-deoxy, wala yung OH group. There are two hydrogens on carbon number 2, kaya 2-deoxyribose. Okay? So in terms of sugar component, yan yung pagkakaiba ng DNA and RNA. Next, the bases. Let us look at the bases. Okay? So the nitrogen containing base is one of 5 types. So there are 5 types. Okay? So pag tinignan natin yung first set, okay, of the nitrogenous base, we see the similarity in their structure. They are made up of one ring, cyclic ring, okay? And of course, six members in a ring. And of course, we also see nitrogen. So lahat naman may nitrogen and oxygen. So the structures of these bases, what are the bases? Cytosine, letter C, uracil, letter U ang representation, thymine, letter T. They are similar to what? To the parent compound. What is the parent compound? Pyrimidine. So this is the structure of pyrimidine. So lahat sila merong ganitong cyclic structure. Okay? And the second type, okay, the second set, we have adenine, letter A, guanine, letter G, capital. Okay, if we look at their structures, they're made up of two cyclic structure fused together. And similar sila with the parent compound, purine. Okay? So now, um, DNA has the following or contains the following bases: adenine, guanine of the purine type, okay, and then cytosine and thymine. Whereas RNA contains meron ding A, G, and C. Kaya lang ang pagkakaiba now, okay, RNA contains uracil. No thymine in RNA. Okay, so let's go back to the previous. Okay, so for DNA, merong um, cytosine and thymine. The uracil is in the RNA. So dito sila nagkaiba. Okay, now, next, if now these bases are connected to the monosaccharide, okay, to the sugar component, then we call this nucleoside. How do we name the nucleoside? Okay, how do we name the nucleoside? Meaning to say, you have now this bases connected to the sugar. And the connection, na-mention ko kanina na the anomeric carbon, that anomeric carbon is connected to the nitrogen of the bases. Ito yan. Dito, o kaya dito. Yan. Okay, now, so if the nitrogenous base is derived from pyrimidine, the name of the nucleoside ends with idine. Okay, idine. So look at the suffix, yung last letter of pyrimidine, idine. Okay, so gawin natin dito. So for cytosine, if a sugar is attached to it, then it becomes citidine. Okay? If a sugar is attached to uracil, then it is now uridine. If a sugar is attached to thymine, then it is thymidine. Okay, or thymidine. Now, if the sugar is a ribose, okay, so meaning to say it is in the RNA, so simply we call it citidine, okay, uridine. Okay, now if it is connected to a deoxy ribose, we have to use the prefix deoxy. So we know that in the DNA, cytosine is present, right? So cytosine connected to deoxyribose. Lalagyan natin ng prefix na deoxycytidine. Ito rin, deoxythymidine because thymine is present in DNA. Walang uracil sa DNA, right? Okay, so that's how we name the nucleoside. Depende kung where the the base is derived, no? Ano yung parent compound. If the parent compound naman is from purine, okay, the suffix we add is osine. Osine. Okay? So, for adenine, it becomes now adenosine. Ibig sabihin, the adenine joined to the monosaccharide, we call now that nucleoside as adenosine, yung isa naman guanosine. Ngayon, kung nasa RNA yun, ribose. So, as in, adenosine, guanosine. Pero kapag sa DNA, because adenine and guanine are all present in DNA, then we have to add the prefix deoxy. Deoxyadenosine, deoxyguanosine. Okay? Did you get that, class? Nakuha nyo how to name the nucleosides. Okay? So now, let us move on. Okay, so it says here, a nucleoside is formed by joining the anomeric carbon of the monosaccharide, which is carbon number 2, okay, with a nitrogen atom of the base. Na ipakita ko sa inyo kanina kung alin yon. To name a nucleoside derived from pyrimidine base, use the suffix idine. Okay, tanda, pyrimidine, idine. Now, to name a nucleoside derived from purine base, use the suffix osine. And for deoxyribonucleoside, meaning to say nasa DNA makikita, we add the prefix deoxy. Okay, so may mga examples na tayo kanina, no? Okay, next now. Okay, so here it shows the connection, the bond. Okay, so carbon number 2 and the nitrogen of the nitrogenous base. This is it now. And so anong tawag dyan sa linkage or bond na yan? Capital N glycoside or glycosidic linkage. Oops, familiar. Parang narinig na namin yan, ma'am. Yes, sa carbohydrates, di ba? The linkage between the monosaccharides in a carbohydrates ay tinatawag na glycosidic linkage. Dito rin, glycoside or glycosidic. Kaya lang we have to include a capital N like this which refers to the nitrogen there in the nitrogenous base. Okay? Now, so this is cytidine. So na ano natin kanina, 'di ba? Nagbigay na natin kanina yung one example of a nucleoside. Okay, next. If you have in the DNA a deoxyribose that is connected to a nitrogenous base which is of purine, from the purine, adenine. So the name now would be, dadagdagan mo ng deoxyadenosine. Okay? Can you follow? Can you follow? Nasa structure tayo, mag-a-appear yan, ha? Okay, next now. Punta na tayo sa nucleotides. If we join now, or if we add a phosphate group to the left side of the sugar, connect it to carbon number 5 of sugar, then we have now the complete nucleotide, mononucleotide, having now the three components. You have the phosphate, the sugar, and the nitrogenous base. So how do we name it? Or before we name it, says, nucleotides are formed by adding a phosphate group to where? To the 5' OH of the nucleoside. Ito yung 5'. Okay? Ito yung fifth carbon. Okay, now, how do we name it? We simply add the word monophosphate because there's one phosphate group added to carbon number 5. So, the name kanina ni nucleoside, cytidine, and then add 5 prime dash monophosphate. So, this is now a nucleotide, specifically a ribonucleotide because we have ribose as the sugar. Okay? And then, of course, ang haba ng pangalan, there is an abbreviation for this. The abbreviation is capital C, capital M, capital C. P, the C stands for cytidine, the M for mono, and P for phosphate. Okay? So we have now, okay, ayun na. The resulting nucleotide, inulit lang dito. The name cytidine 5 monophosphate is abbreviated as capital C, capital M, capital P. Meaning to say, kapag yan ay nag-appear sa quiz, do not write this in small letters. Okay? Next. So, here, what is shown here is the formation of the bond. Okay? So dito ang tanggal ito, the OH, and then kabit ito doon. Okay? This is for deoxy. An example, a deoxyribonucleotide. Okay, so this is deoxyadenosine. Okay, what else? Okay, so now, how do we name if we have a deoxyribonucleotide? So this is yung pangalan ng ano kanina, ng nucleoside, deoxyadenosine. And then we only add 5 prime dash monophosphate. The abbreviation is for deoxy, yung small, yung D doon, small d. Okay? And then capital A, capital M, capital P. So this is the abbreviation for deoxyadenosine 5 monophosphate. These abbreviations, we'll see them more pag nandun na tayo sa last two modules, bioenergetics and metabolism. Okay? Next. And so, if another phosphate group is added to the 5 prime carbon, so you have now two phosphate groups attached to this, then this is now a diphosphate. ADP, familiar na, di ba? Because if you add another phosphate group, we have three phosphate group, that's ATP. ATP and ADP, very common nating nakikita because they can be interconverted to each other. So adding a phosphate to ADP, it becomes ATP. Removing a phosphate group from from ATP, it becomes an ADP. And so more of that, okay, will be discussed pagdating do sa last two modules, and they are what we call um, high energy molecules because breaking the bond here, okay, release a lot of energy. So kaya kapag ka nandun na tayo sa metabolism, what we count ay how many ATPs are formed when a single molecule of, for example, glucose is metabolized. Okay, so, and also, for example, kapag meron tayong a fatty acid, okay, one fatty acid, and it is metabolized, okay, to give energy, we determine that amount of energy by counting the number of ATPs that are synthesized or from the metabolism of that molecule. Okay? Next, ano pa ba? Okay, ayun na si ATP. ATP is an example of a triphosphate. So adenosine 5 prime triphosphate is given the abbreviation ATP. Okay? Of course, no, yung T comes from the phosphate. Capital P sa phosphate. Then, ano pa? Okay. So, nucleic acids, DNA and RNA are polymers, okay, of nucleotides. So here we have two nucleotides. And how are they joined together? They are joined by a linkage called phosphodiester linkage. Ito yun. So this is the phosphate group of the second mononucleotide. Okay? And this is linked to carbon number 3 of the first nucleotide, carbon number 3 of the sugar component. Yan. So this now becomes the phosphodiester linkage because if para siyang ester, di ba? OR, OR. Okay? Kaya phosphodiester, dalawang ito, no? An ester group. Okay, kaya phosphodiester linkage. This is the type or the name of the bonds joining the two mononucleotides. Okay? So if you add more nucleotides, then you have now a polynucleotide. The nucleotides are joined by that phosphodiester bond. So it says here, a polynucleotide, okay, contains a backbone, okay, consisting of alternating sugar and phosphate group. And the identity and order of the bases distinguish one polynucleotide from another. So therefore, there is again what we call a primary structure. Just like sa proteins, yung sequence ng amino acid, that's the primary structure. So meron din ganun sa nucleic acid. Okay? And a polynucleotide has one free phosphate group at the 5' end and one free OH group at the 3' end. Okay. In the DNA, the sequence of the bases carries the genetic information of the organism. So let us look at an example of a polynucleotide. Ito. Okay. Nasaan yung backbone? Okay, tingnan natin. The backbone is this one. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay? And so here, clearly we can see the phosphodiester linkages joins what? The 3' carbon of one nucleotide to the 5' carbon of the next nucleotide. Yan. 3, 5, 3, 5, 3, 5. And a polynucleotide has one a free phosphate group at the 5' end and a free, pag sinabing free, hindi nakakonect, walang connection, di ba? OH group at the 3' end. Okay? Next. Napakasunod. Okay, so now this one, the chain, the polynucleotide, can be abbreviated using the names or the one-letter symbol of the bases, nitrogenous bases. So, doon sa sa kabila, tingnan natin yun cytosine, adenine, thymine, guanine, those are the nitrogenous base component of the nucleotides. So, abbreviation, we use the letter C, A, T, G, which is shown in the next slide. Ayan, C, A, T, G. And here, ito yung sugar phosphate. Okay, the sugar phosphate. Siyempre ito yung sugar phosphate. Okay. Sugar phosphate, sugar phosphate, sugar phosphate backbone. Okay, so this polynucleotide would be named ATG, reading from if we start reading from here, 5' end to the 3' end. Okay? So that's how we read. That's how we give the name of the polynucleotide. We start from the 5' end to the 3' end. Okay, next. Now, okay, what is the structure of the DNA? No, the DNA structure is double helix. It is called double helix. No, and kanina, okay, so the DNA model was initially proposed by Watson and Crick in 1953. DNAs consist of two polynucleotides. So here, another differentiation between DNA, okay, and RNA. Kanina RNA, sabi natin, is just one polynucleotide, one strand, and it contains thousands of nucleotides. Ang DNA has two strands, two polynucleotide strands, okay, that wind into a right-handed double helix. So nakatwist yun to each other, okay, forming a double helix, parang spiral stair, 'di ba? Yung ano no, yung stair na ano, spiral. Okay. So, the two strands run in opposite direction. One runs from 5' end to the 3' end. So, from the top, magsimula tayo sa top. So, yung isang strand is running from 5' end to the 3' end. The other one naman nakayakap niya runs from the top again, 3' end to the 5' end. This way now, class, okay, what we see ay the sugar phosphate uh, groups, yung backbone, ay nandun sa outside of the helix. And the bases lie on the inside. And the bases, class, the bases, no, this way of arrangement of if from one strand you have a pyrimidine base, on the next strand, nandun naman ay purine base. Yung dalawa na yon form a band, okay? Forms a band. And that stabilizes the double helix structure of the DNA. So, ito yun. Dito natin makikita sa susunod na slide. Ayan. So, ito yung backbone. Sugar phosphate backbone. Kasi dalawang, ano yan, dalawang polynucleotide. This is the first one and this is the second one. So, ito yung parang um, yung sa hagdanan, sa stairs, di ba? Yung humahawak sa mga baitang. Okay, so yun yung we call the backbone. Ito yung the steps of the ladder. Okay, the bases. And if here, for example, G is guanine, which is a purine base. Doon sa kabilang strand, you have a pyrimidine base. And what holds these two together are very strong bonds, hydrogen bonds. Okay? And if there, we also see that if there is a G, it is always bonded to C. If in the other you have a T, okay, the other strand naman, there is a. So ito yung base pairing. Okay? That's always no the base pairing. C pairs with G and T or A pairs with T. Okay? So, ano yung mga hydrogen bonds? Yan. So, G-C pair, guanine and cytosine. Okay. They are being held together by three strong hydrogen bonds. You see the broken lines? Yung broken lines na yan yung hydrogen bonds. Very strong. Okay? And then, yung adenine and thymine naman, they are being held to each other by two strong hydrogen bonds. Okay, so these interactions are responsible for this double helix structure of the DNA molecule. Okay, now next. And then it says here, the bases always line up so that a pyrimidine derivative can hydrogen bond to a purine derivative on the other strand. Thus, there are complementary base pairs. So we call this complementary base pairing. They always hydrogen bond together in a particular manner. Adenine pairs with thymine with two hydrogen bonds to form an A T base pair. Cytosine naman pairs with guanine using three hydrogen bonds to form a CG base pair. Okay? So, if we know the sequence of the bases in the first strand, we can give the sequence of the bases in the second strand, which we will do as a sheet work later. Okay? Next. So, yon. The information stored in the DNA, okay, is used to direct the synthesis of proteins. Ano yung information stored na yon? This refers to the bases, nitrogenous bases, because ay consecutive tatlong nitrogenous bases will define or will give a specific amino acid. Okay, now, nasan na tayo? For us to learn how information are transferred or is transferred from DNA, okay, to RNA, to the formation of um, proteins, synthesis of proteins, o kaya during replacement of worn out cells, then we have to understand three key processes. What are these? Replication, transcription, and translation. By the way, class, are you still here? Are you still with me? Are you listening? Of course, I cannot ask, no, hindi ako pwedeng magpa-recitation. Siyempre kasi I have to discuss muna. We have time for recitation. Okay, thank you for answering. Now, okay, tinatawag din to, no, the central dogma of protein synthesis. So let's start with what is replication? The process by which DNA makes a copy of itself when a cell divides. Okay, so minsan 'di ba, when new cells are produced, so merong cell division, blah 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 blah. Okay, and then now, when of course sa replication, eksakto kung ano yung parent DNA, it will form the same daughter cells. Okay, daughter DNA. Then, if the information must be passed on so that uh, certain proteins is synthesized, then kailangan muna yung information dun sa DNA ay ma-pass to an RNA. So that is transcription. Okay, transcription is the ordered synthesis of RNA from DNA. So buhat kay DNA, the RNA will be synthesized. And so whatever is the genetic information stored in the DNA, it is passed on to the synthesized RNA. Okay? Now, so nasa RNA na. Yung information na nasa RNA na yun, i-convert na to protein. So that is translation. Translation is the synthesis of proteins from RNA. The genetic information determine the specific amino acid sequence of the protein. And that genetic information class, yun yung sequence of nucleotides or the nitrogenous bases. Okay? Next. Now, so, this is the central dogma of protein synthesis. So, during, when parent DNA, converted to daughter DNA, we call that replication. DNA to RNA, transcription. RNA to protein, translation. Okay, now, isa-isahin natin muna. During replication, what happens during replication? Okay, tignan muna natin how we we give, no? The sequence of the bases doon sa daughter DNA. Okay, so let's start here. The original DNA molecule. Okay, so the original DNA molecule, alam natin sa nasabi na kanina na it is made up of two polynucleotide strand twist around each other, and the one strand is running from 5' end to the 3' end. Yung isa naman running from 3' end at the top going to the 5' end. So, let's start with the first strand. Ito, the one that is starting from 5' end. So, ito yung sequence ng bases niya. A, T, T, C, G, A for the first strand. And they are, both strands are in colored blue. Okay? Now, yung kayakap niya, itong the second strand, which runs from 3' end, okay, to the 5' end, ito naman yung sequence ng bases. How do we know the sequence? Just the complementary base pairing. This is A, then it is paired with T, thymine. This is T, so the second strand would be A. This is T sa first strand, the second strand, that would the base would be A. This is C for the first strand, then G for the second strand. This is G for the first strand, C do sa second strand. A for the first strand, then sa second strand, it should be T. So always, ang base pairing na followed is A, P, and G, C. Okay? So that's it. Now, so, during replication, mag-unwind ito. Mag-unwind yan. Okay? Pag nag-unwind yan, class, then, okay, each strand, each strand will serve as a template. Okay? So, let's have muna the strand, no? The one. Okay. Yung isang strand, ito. The one that is running from the 3' end, ito yon. Yan, yan. Yan yung kulay blue. Okay? Yung kulay blue. That is from the parent. 'Di ba? Parent DNA. Ito yon. Which is colored blue here. So that's T, A, A, G, C, T. Ito yung strand number 2 sa parent DNA. So, what we do now, to give to give the sequence of the bases in the new strand, which is red in color. Ito, red in color. So we just give the base pairing. T in the parent strand. So in the new strand, daughter strand, it would be A. Okay? This is A, then this is T. A, this become T. G, this is C. And so on and so forth until you produce the new strand. Okay? So, ito na ngayon yung magkayakap na 2 polynucleotide strand. Okay? So, this is one daughter DNA. Another daughter DNA is if we have strand 1 which runs from 5' end to the 3' end and it is blue in color. Okay, so how do we give the sequence of the bases in the new strand which is red in color here? Ayun, we just give the base pairing. Okay? So hanggang here you have A. So the first base would be T in the new strand hanggang makarating tayo dito. Now, let us compare this new daughter DNA to daughter DNAs with the original one. Okay, di ba? Para A, T, TA, parehas na parehas. Di ba? So that's replication. And this is called semi-conservative because one strand from the parent ay nandoon sa daughter. So conserve, semi-conservative. Okay, yon ang ibig sabihin. Do you understand? So, given the sequence of nucleotides, no, in the sequence of the bases in a polynucleotide, you can now give the other strand. Okay? What else? Okay. So, ayan yan. No. Okay. Aha. Sandali konti lang. Okay. Okay, so how does replication takes place? Okay, before replication, ang itsura, the shape of the DNA, as we know, is a double helix like this. Okay? Now, the first step, tignan natin ha. Okay. Here, what is shown here is, uh, of course, ano no, the first step would be unwinding, yun. Unwinding of the double helix to expose the bases. Okay? Pag nag-unwind ngayon, there is a creation of what we call replication fork. A replication fork forms as the two strand split apart, okay, exposing these bases. And then each of these strands serve as a template. Okay? Next. And so, one of the strand is called a leading strand. And the other strand is called a lagging strand. Lagging strand. Bakit? Anong kaiba ng dalawa? Yung leading strand kasi, when it is read, okay, um, ah, continuous ang pag-replicate, okay? Yung pag-form ng other strand. Yung isa naman, the lagging strand, it is replicated in segments, putol-putol. Okay? Why? Okay, so let me read this to you. The first step in replication is the unwinding of the DNA helix to expose the bases on each strand. Nandun yun sa ano eh, sandali lang ah. Okay? Okay. Ayan. Andito sa kabila. Ayan, yan. Ito. Okay? So, the first step in replication is the unwinding of the DNA helix. Unwinding. To expose the bases on each strand. So, yan. Expose yung mga bases in each strand. Unwinding occurs at many places. So, hindi lang sa isa. So, pwedeng sa iba-ibang ano, no? Okay? Along the helix, creating bubbles, okay, where replication can occur. The point at which unwinding occurs is called the replication fork. Here it is. Unwinding breaks the hydrogen bonds that hold the two strands of the double helix together. Okay? Once bases have been exposed on the unwound strands of the DNA, the enzyme DNA polymerase catalyze the replication process using the 4-nucleoside triphosphate derived from A, T, G, and C. And you will understand it more when you watch the video. May videos na nandun sa module. Paki-panoorin na lang. Okay? Mm. And so, okay, yung isa, tignan natin ito ha. Okay? The reading, okay, it says here, replication proceeds in the 3 to 5 direction. 3 to 5 direction class. Ito yung original strand, di ba? Ito, this one. So, nag-unwind yan. Ito yung, the reading is always start from the 3' to the 5' direction. Okay, so itong strand na to, which is being read from 3' to the 5' end, the replication is continuous. Ito yung replicated strand. Oh. It is being formed continuously. Okay? Whereas the other strand, since dapat the reading is always from 3' end to the 5. So yung 3', ito kasi yung 3' end nitong isang strand. Ito yung 5' end. So from 3' end habang bumubuka ito, okay, ang pag-replicate is from this direction going here. 'Di ba? From this direction. Ngayon, pagbubuka ito, so doon na naman. So kaya putol-putol, ibig sabihin. So, the replication is in fragments, also called Okazaki fragments. You'll see that in the video. Okay? So, of course, there are enzymes no, that are responsible. DNA polymerase, ayan, okay? And then, nucleoside triphosphates, these are available in the nucleus na ginagamit para to give the complementary base pairs. Okay? So that's how replication happens. So it says the identity of the bases on the template strand determines the order of the bases on the new strand, which we have discussed kanina, how to determine those sequence of the bases. A must pair with T and G must pair with C. And a new phosphodiester bond is formed between the 5-phosphate of the nucleoside triphosphate and the 3'OH of the new DNA strand. Replication occurs, tandaan nito, replication occurs in only one direction on the template strand, whether it is the leading strand or the lagging strand. It is from the 3' end to the 5' end. Okay? So the new strand is either a leading strand growing continuously or a lagging strand growing in small fragments. Okay? Naintindihan, class? Do you understand? Hello? O kaya ang sarap na ng tulog nyo? Okay, thank you, Mr. Sobramonte, for answering. Okay, next. Mhm. RNA. Okay? So, let's talk about RNA muna. Kanina halos karamihan about DNA, no? Although, naiko-compare si RNA with DNA. So, there are important differences between DNA and RNA. Inulit dito. In RNA, the monosaccharide is ribose. Okay? Ano pa? Then, nitrogenous base. The thymine, although I have already mentioned this, the thymine base is not present in RNA. Instead, it is uracil. Okay? You have uracil in RNA. In RNA, nasabi na natin, is a single strand. Tapos, smaller than DNA. And there are three types of RNA molecules. These are the ribosomal RNA, given the abbreviation small r, capital N, capital A. Then the messenger RNA naman, small m, capital R, capital N, capital A. And the transfer RNA, capital T, small t, capital R, capital N, capital A. Okay? So, if nag-appear sa quiz, do not change the abbreviations. Okay? Next. So, let's differentiate one from the other. What is ribosomal RNA? Ribosomal RNA, yung mga ribosomes na nandoon sa cytoplasm. Yun ang site kasi ng protein synthesis. So ribosomal RNA provides the site where polypeptides are assembled during protein synthesis. Okay? The messenger RNA, this one carries the information from DNA to the ribosome. Meaning to say, the mRNA is the one that is being synthesized in the nucleus. Okay? Wherein yung information in DNA is transferred to RNA, and the specific RNA that is synthesized there is the messenger RNA. Okay? Now, when the messenger RNA has been synthesized, it will go out of the nucleus, go to the ribosomes, okay, where you have the ribosomal RNA there, providing the site for, no, ano yun, yung assembly of protein, no, synthesis. Then, pag nandun na siya, okay, the transfer RNA naman, tRNA, these are the one that brings specific amino acids to the site, to the ribosomes for protein synthesis. They carry specific amino acids, and these specific amino acids are the one that is coded by the bases, three bases present in the mRNA. Okay? So, transfer RNA is drawn as a clover leaf shape with an acceptor stem at the 3' end which carries the needed amino acid do sa 3' end and an anticodon which identifies the needed amino acid. Okay, so tingnan natin ito. Okay, this is how, no, this is a representation of transfer RNA, clover leaf daw representation. So, okay, RNA single strand. Yan, single strand lang yan. Kaya lang syempre, to have this structure, meron syempreng ano dyan. Tawag dito is hydrogen bonds between the bases, right? Now, the important ano dun is, okay, so from the 5' end to the 3' end, ayun. Doon sa 3' end, this end carries the amino acid being coded by the mRNA. Diyan yung amino acid, dala-dala niya. Doon sa site, dadali niya doon sa ribosome. And this portion naman ng anticodon, this one naman, when this transfer RNA goes to the site, this anticodon bonds with the codon na nandoon sa mRNA. Once a bond is formed between the two, then this will be released, the amino acid is released, and then these amino acids are assembled to form the protein. Okay? So dinidiscuss pa lang natin yung features ng mga RNA. Okay? So there is an anticodon portion and a acceptor stem. Okay? Where it carries, yung nagki-carry ng amino acid. So look, no, nakalagay yan, hydrogen bonding between complementary base pairs is responsible for this shape or structure of the transfer RNA. Okay, next. So, nasaan na ba tayo papunta? Now that we have learned the features of RNA, we're now on the second key process, which is transcription. The information in the DNA is now passed on to the RNA, specifically mRNA. So, transcription is the synthesis of mRNA from DNA. Since DNA is nasa nucleus, so therefore, sa nucleus na ang site of synthesis ng mRNA. So again, the DNA splits into two strands. The template strand. Pag sinabing template, yun ang gagamitin para matranscribe yung mRNA, masynthesize yung mRNA. The other strand, kasi double strand ang DNA, di ba? The other strand is called informational strand, which is not used. Okay? So, again, the transcription proceeds from 3' end to the 5' end of the template strand. Transcription forms a mRNA with a complementary sequence. So we still follow complementary base pairing to the template DNA strand and an exact sequence, yes, an exact sequence of the informational DNA strand, which we will see later. The difference between mRNA and the information DNA strand is the base U replaces T on mRNA. So, mas maintindihan natin yan mamaya pagka may example na. Okay, so let us look at this now, yung sa transcription. Okay? So, again, what happens is, so, at start, there would be unwinding of the double helix. Okay? And during unwinding, may expose ngayon. So, unwinding, masisira yung hydrogen bonds that hold the strand together, right? Exposing the bases. One strand is called an informational strand, which is not used. The other strand is the one that is used to synthesize the mRNA. And a certain portion of it lang, certain portion lang of the whole DNA molecule, which called gene, yun yung nata-transcribe. Okay? And it says a gene that codes for a specific protein. So, what happens now during unwinding? Okay? So, ito yun, no? Ito yung non-template strand. So, pinalaki. So, you have the non-template strand. This is the template strand. Okay? And then it says, the direction of the transcription is okay, from 5 prime n to the 3 prime n. Pero what is being read in the template is from 3 prime n to going to the 5 prime n. Okay? So, ito ngayon yung na-transcribe na mRNA, which is orange in color. So, ito na si RNA. So, isa lang, isang strand lang. Then, the DNA will reform. Okay? The DNA double helix reform. So, once this is created, lalabas na yan doon sa nucleus. Of course, there are modification which you will also see in the video. So, please watch the video para makita nyo yung ganda na ang how these things happens. Okay? So, next. Okay, so ito yung examples, okay, ng transcription. From the template strand of DNA, write out the mRNA and informational strand of DNA sequence. Okay, so ang pinapakita dito is a DNA, the template strand. So the DNA is made up of two polynucleotide. Okay, two strand. One strand, which is the template strand, okay, runs from the 3 prime end to the 5 prime end. Okay, ito yung sequence ng bases. C, T, A, G, G, A, T, A, C. Cytosine, thymine, adenine, guanine, blah, blah, blah. Okay? Now, the mRNA that will be transcribed from this template strand, which is being read from 3 prime end to the 5 prime end, ito. And the mRNA now, the transcribed mRNA now goes from 5 prime N, opposite of this, 5 prime N to the 3 prime N. So, we just give the complementary base pair. So, there is C in the template strand, G ang kapartner nun, ka-complementary nun. There is T in the template strand, then A in the mRNA. You have A in the template strand, A pairs with T, sabi natin. But then, since there is no thymine in RNA, but instead uracil, for A now in the DNA strand, template strand, it will be uracil in the RNA. Okay? Am I clear on that, class? Nagkakaintindihan tayo doon na wala kasing thymine or T sa RNA, ha? Okay? So may mga ganitong questions both in your postas and in the final exam. Okay? And then G pairs with C, G pairs with C. Remember, A, G, C are both in the RNA and DNA. Nagkaiba lang sa T at saka sa U. Okay, now, meron uling A sa DNA. So, pagdating sa RNA, it is U ang ka-pair niya. Then next, there is T in the DNA template strand. So therefore, T pairs with A, so you have A adenine present naman yan sa RNA. Okay? A pairs with uh, uh, U and then C with G. Okay? So, yan yung na-transcribe na mRNA. So, yung information, which are the sequence of bases, yun yung na-pass on kay RNA. mRNA to be specific. Now, ano naman yung informational strand na sinasabi dito? What is being asked here is you give now the other polynucleotide strand na kayakap ni template strand. 'Di ba dalawa 'yon, double helix, 'yun ang dalawang polynucleotide strand 'yon. Yung template strand at saka informational strand. So what we do is we simply give the base pairing of this, okay? Tapos, syempre, direction ay magkaiba. This is from 3' running from 3' end to the 5' end. So therefore, the other strand na kayakap, okay? We're talking about the DNA molecule, ha? So 5' end. So C, ka-complementary niya is G. T, A. A, T, G, C, G, C, A, T. T, A, and the next A, this must be T. Typographical error. Walang uracil sa DNA. Di ba? So this must be T and then G. Okay? Am I clear on that? Now, if we compare informational strand with mRNA, they have the same base pairs except that kung U sa kung U sa RNA, T naman 'yon sa informational strand. Do'n lang sila magkakaiba. Okay? Am I clear, class? Hello? Kaya pa ba? Kaya pa ba? It's 8:32. I will stop. Doon na tayo we will start with the genetic code next meeting. Here, we read a three bases, we call it codon, dun sa mRNA. Tapos, hahanapin natin dito sa, yun, ito oh, the genetic code. Hahanapin natin. Ito yung mga four bases in the mRNA. U, C, A, G, okay, yan. Hahanapin natin yung equivalent na amino acid being coded by those three bases. Okay? Am I clear on that, class? So, dito tayo mag-start next meeting. Okay, pwede ba? Mag-rest na tayo. It's already 8:30 para naman hindi na masyadong saturated yung inyong Okay? Hello? Are you still there? Bakit naging 36 na lang tayo? Okay, save username. Yes, Miss Cruz, you have you have question, Miss Cruz?
Ma'am, may concern lang po ako regarding sa evaluation.
Yes, yes.
Nung binuksan ko po kasi yung site, hindi po siya
Oo,
lumalabas.
gano'n?
Nakalagay
Ah,
po is a timeout occurred daw po.
puwede bang picturean mo 'yan, tapos i-send mo sa'kin para ise-send ko doon sa evaluation team, yung sa department that is in charge of that, puwede? Ha?
Sige po ma'am,
Kasi
thank you po.
kasi may deadline sila na 24, 'di ba? So dapat ano 'yan, ha? Sa akin din po ma'am, ayaw po mag-load. Okay, puwede ba i-message niyo 'yan and then I will just copy that and send to them. Okay? Thank you for that, okay? So if you have no more question, I hope you learn, I hope you understand. Ako rin kasi class, kahit na paulit-ulit kong tinuturo, pero nakakalimutan ko ulit. Pero pag binabasa ko, naalala ko naman. And I'm happy kapag na-explain ko ng maayos sa student at naiintindihan naman ng student. Yes, Miss Merced? Miss Merced? Yes, Miss
Ma'am,
Merced?
okey na po pala, kaka-load lang po ngayon sa'kin, lumabas na po siya.
Okay na, lumabas na. Okay,
Opo.
sige.
Thank
Mm.
po.
Basta itago nyo lang muna, i-save nyo lang muna yung proof, ha? Kasi ang dami ko talagang ginagawa, class. Hindi na ako humihinto ng kaka-check. Ha, please? Okay? Thank you. Thank you. Good morning po. Yung quiz postas po ba sa Saturday sa about enzyme lang? Wala pa akong gagawing quiz on Saturday. Okay? No quiz yet. No postas yet. Okay? So continue tayo ng discussion on Saturday. Kailangan nating matapos ang nucleic acid. Okay? Okay. Thank you everyone. Thank you. Thank you. Yeah.